So, a bit about God's amazing creation. This person writes, I work at the doggy daycare, and this was them all going out to the big play yard. Look at the joy. Look how happy and excited they are. Oh, yes, and look at, I think it's a Brittany Spaniel tripping over the camera at the end. Shame.
Everybody loves a picnic. With that straw-like proboscis, this fly is going to slurp your lunch, if you let it. Add in a little regurgitated liquid, and it turns your burger into a shake. Think you can take it on? A fly is a formidable opponent. Its hairs and antennae feel the air move as you approach. And just look at its bulging eyes. It can see you coming from nearly every angle. Its eyes and tiny brain process information 10 times faster than ours. To a fly, we appear downright sluggish. In the air, it's in its element. Check out those razor-sharp turns. Sticks the landing. Gone in a flash. If you look at a fly really close up, you'll see how it does it. Check out that club-shaped part moving back and forth, right below its wing. It's called a haltere. It's what makes those athletic moves possible. Scientists have identified 200,000 kinds of flies. They all have halteres. Haltere means dumbbell in Greek. See that dumbbell shape on this crane fly? Flies have 2 wings and a pair of halteres. Most other insects have 4 wings. As the fly turns, its halteres sense the movement. In a split second, neurons at the base of the halteres send information to the fly's muscles to steer its wings and keep its head steady. When researchers remove a fly's halteres, it can no longer control its flight. It loses all sense of where its body is in space. If it gets inside your house, its halteres will help it do a fly's signature move, the ceiling landing. It hangs there with tiny hooks and sticky pads on its feet. The pads, called pulvilli, have microscopic hairs that excrete liquid. That liquid sticks to the surface under pressure, kinda like suction. Despite the fly's arsenal of slick tools, next time you go hunting, here's one trick you can try. A fly struggles to see objects that are still. So if you go very slowly and then pounce, you might stand a chance.
The human brain, one of our most complex organs, allows us to move, feel, think, and learn. Inside the brain, a dynamic network formed by billions of nerve cells processes information via electrical signals. Each neuron receives information from thousands of other neurons via multiple processes, called dendrites, and transmits information via a single axon. At specialized contact sites called synapses, electrical signals trigger the regulated release of neurotransmitters stored in synaptic vesicles. These chemical messengers bind and activate postsynaptic receptors, initiating a new electrical signal in the next cell. But how does a presynaptic cargo, for example, made in the cell body, end up at the right place within the cell? For this, neurons use active transport to sort specific synaptic proteins between the axon and dendrites. The microtubule cytoskeleton is important for this polarized cargo transport since it provides the roads for molecular motors. Microtubules, built up from tubulin subunits, are dynamic structures. Two types of motor protein, kinesins and dynein, transport cargoes along microtubules. Adapter proteins help to couple the correct cargo with the right motor protein. Kinesin and dynein move in opposite directions. Dynein moves towards the minus end, whereas most kinesins move towards the plus end. Therefore, the orientation of the microtubules helps to direct polarized transport. But many more levels of regulation exist to ensure proper sorting of cargoes. Various modifications of microtubules provide additional cues for motor proteins. Kinesins themselves are regulated by autoinhibition to prevent unnecessary activity. Attachment to a cargo or modifications of the motor protein releases autoinhibition and activates the kinesin. Multiple motor proteins can be attached to the same cargo, causing bidirectional movement. Finally, reaching the right destination, the cargo is delivered, and in this case, released at the presynapse.